Hi, let's get started talking about cells. It's time to begin. So our chapter 1 is called Cells, the Fundamental Units of Life. You've heard that phrase before. What do we mean when we say cells are the fundamental units of life? Well, as you know, cells are the smallest entities that have the properties of life. This is the first statement of cell theory. It was coined over 150 years ago when people really didn't even know what cells were. So we know a lot more now, and there's still a lot more to be learned. When we study cell biology, we're studying the structure of cells, the structure of the different components of cells, the organization in which things are arranged, various functions that cells can accomplish, and also how cells behave in a community of other cells, whether it's within a multicellular organism or as individual unicellular organisms. This chapter has 5 main sections to it. The 2 sections marked with an asterisk are the ones that we'll cover in this video. So all cells have that common property of being alive, but yet cells are extremely diverse. Some cells are huge, others are very, very tiny. Some cells have many different components, and others have sort of a minimum level of components, just enough to be alive. Some cells have different shapes. The images that you see across this slide give you some idea of the range of shapes that you may see, but there are so many other shapes as well, and the shapes, of course, directly relate to the functions of these different cells. Some cells require only carbon dioxide, water, and light in order to be able to survive. Other cells need complex biomolecules in the form of nutrients to be brought in from the exterior of the cell, in addition to things like oxygen, or maybe not oxygen, maybe sulfur. For some cells, oxygen is poisonous, and so the chemical requirements vary tremendously as well. However, underlying it all, that property of life demands that living cells all exhibit a chemistry that's fundamentally very, very similar. The molecules that we call biomolecules that do the jobs of a cell, and the chemical reactions that happen in the cells are amazingly similar, whether you're talking about a unicellular prokaryote like a bacterium, or if you're talking about an insect, a plant, a mammal, or some other type of organism. What we call the central dogma of molecular biology is portrayed here. That is, that the DNA molecules are information storage molecules. That information is key, it's vital to everything that happens in the cell. When a cell is going to replicate, it makes copies of its DNA to pass on to its daughter cells. But that DNA information is organized into discrete units that we call genes. These genes individually can be transcribed into RNA molecules, another main type of biomolecule. These RNA molecules provide the instructions for building a particular protein, and these proteins do the jobs that dictate a cell's form, function, and behavior. The chemical reactions that happen in cells are very complex. They're actually organized into sequences of reactions that are interconnected with one another, and they're very highly regulated. They produce cell products, they modify molecules of different types, they allow self-replication, they allow for energy to be converted into ATP energy or GTP energy, other forms of energy that the cell is able to use directly. So the second statement of cell theory is that all of life's processes happen in cells, or they happen as a result of what has taken place in a cell. Because these various life processes require chemical reactions that interact with one another, and that oftentimes can interfere with one another as well, quality control in cells is absolute. So the third statement of cell theory is that cells come from pre-existing cells. To get daughter cells, you have to start with parent cells, pre-existing cells. Scientists have tried for centuries to combine the various types of molecules that we know exist inside cells, put them together, add energy, and yet we can't make a living cell just from the cellular components. Our second section in this chapter deals with microscopes. It wasn't until microscopes had been invented that we were actually able to discover cells. So using light microscopes, we can see some or even many of the components of a cell, but to really be able to see fine detail as we would like to, we need to use electron microscopes. It wasn't until light microscopes were invented, and Van Leeuwenhoek had one of the early microscopes, and he decided to look at pond water. When he looked in the pond water, he saw these fantastic tiny creatures that no one who existed before him had ever seen. He drew amazing, beautiful drawings of these protozoans that he called animalcules. Well, since his microscope, the crude one you see on the left, we've had many technological advancements, so a computer-enhanced light microscope is portrayed in the middle of the row of images on this slide. However, ultimately, no matter how much computer enhancement we have, the limits of a light microscope are going to be associated with the wavelength of light. And so they can't really resolve anything that's smaller or closer together than 0.2 micrometers. Regardless of what kind of microscope you're using, careful attention to detail is going to be very, very important. That's illustrated really well in this pair of views from microscopes, one from 1880 and the other from 1985. So Strasburger, one of the early renowned cell biologists, observed living cells in the root of a plant, and he made these drawings over a period of time of 2.5 hours, looking at these a particular cell as it was undergoing the process of mitosis. Remember, this was 1880. His microscope was not anything as advanced as what we have right now. 100 years later, Kepler observed the exact same kind of cells undergoing the same process, mitosis, over the same 2.5 hour period. What he was able to photograph through the microscope reveals structures and movement and patterns that are virtually identical to what Strasburger saw. So as an observer, Strasburger knew what he was looking for, and he was able to distinguish things very clearly. He had the eye of an expert. This is one of the main things that we're going to be working on when we come to lab this week. When you come into our labs, whether it's this class or another one, and you look at images under the light microscope, you might see some things that look like this. The fern root tip on the left and the kidney tubules on the right, depending on the class that you're using. So in both of these cases, these are prepared slides that have been stained using some common biological stains. The fern root tip cells were stained with a dye, a substance that's attracted to DNA and nuclei. And so the nuclei have a very striking magenta appearance. A different kind of dye was also used that stained the cytoplasm, this blue, teal blue kind of appearance. The cell walls don't take up the dye very well, and so you can see where cell walls are around the cells so that the boundaries are very, very clear. And you can see the organization of the cells in the fern. The kidney tubules on the right were also stained, but with different kinds of dyes, so that the nuclei, which take up the dye very darkly, are seen in this very dark purplish color, whereas the cytoplasm takes up the dye less so, and it's in the pale lavender color. Now, when we're talking about animal cells, we don't have cell walls to be able to form thick, distinct boundaries between the cells. Plasma membranes are extremely thin, and so you have to sort of imagine where the plasma membranes are. You can kind of see hints of where the plasma membrane is in some places between the cells, but each one of the nuclei is in a different cell. The cells are long and cylindrically shaped and oriented in a circular arrangement so that the white spots in the centers of each circular arrangement are the openings where fluid passes through the kidney tubules. There are ways of enhancing what you can see with a light microscope without even using a dye. So if we look at the fibroblast on the left, what we can see is when the lenses are the lenses and the filters are adjusted so that contrast is increased. Interference contrast optics is visualized on the left, so the different depths within the cell are distinguishable from one another due to the high contrast. On the right, different kinds of dyes called fluorescent dyes are used to treat different cell components. They're attracted to specific molecules and they emit light at characteristic wavelengths, which a fluorescent microscope can detect. And detecting those different wavelengths of light gives us different colors. So we have one color for the nucleus, another color for the actin components of the cytoskeleton, and yet another color for the melanin pigments that are the products of this particular cell type. A confocal light microscope we'll talk about in just a minute. So since light microscopes only give us a certain degree of resolution, when electron microscopes were invented, because the wavelength of an electron's path is much, much, much narrower, then we're able to see much more finely into the structural components. And so things that are not visible under a light microscope, like ribosomes, like the endoplasmic reticulum, distinguishing between peroxisomes and lysosomes, other characteristics like that, can only be seen by using an electron microscope. This figure gives us some impression of what we can see at different scales, starting with the upper left, the zone that's basically in a real-life unaided view. And I'd like to take you across looking at each pane briefly. If you look at the upper right, you see what you might see using the scanning lens, the smallest objective lens on a microscope, looking at skin. If you come down lower, you would have a high-power image. The next one, the mitochondria, you probably wouldn't be able to see using our microscopes because they're difficult to visualize. And then you come to the right side of the middle panel. What you see is a section that the entire section is 0.2 micrometers in diameter. Remember, that's the resolving limit of a light microscope. Therefore, because all these ribosomes are smaller than 0.2 micrometers, they would not be able to be seen using an ordinary light microscope or even an enhanced light microscope. The images below there could be seen with an electron microscope until you get to the atoms on the far right of the lower panel, which wouldn't even be seen then. There are several panels in this section of the chapter that give you more information about microscopes. We'll be talking about this in lab as well. So on the left, you can see the lenses in their organization in a conventional light microscope. You have some more information about optics and dyes and fixing of samples. This shows us a fluorescent microscope that has a beam-splitting mirror, which is the key to fluorescence microscopy. Some images that you can see using fluorescent probes, especially a confocal scope, which is sort of like a CAT scan as compared to an X-ray, giving a 3D image. Super resolution fluorescence microscopes can show us more than we can see with anything else. These are latest inventions and they're wonderful for looking at living cells as they undergo processes. We can't see living cells using electron microscopes, but the beautiful images that we can see at very high resolution are usually from a transmission electron microscope, as we see here, whereas a scanning electron microscope gives us beautiful images with great detail of the surfaces of cells.